colleagues, we are now going to welcome as focus our research on tumors resistance to immune rejection. And to address this challenge, together with her team, she's been investigating alpha-2 AR agonists and their anti-tumor effects. President, the speaker we are going to give the floor to is going to present very promising results. Thank you.
Le docteur Zhu est docteur en sciences et a obtenu en 2013 un doctorat en génétique humaine à la UCLouvain. Elle a ensuite rejoint l'Institut de Duve et plus particulièrement le laboratoire du professeur Benoît Vandenheim, qui est un des membres de notre compagnie. Elle dirige depuis 2017 son groupe de recherche de manière personnelle. Et elle a été choisie parmi 19 candidats. Elle est effectivement la toute récente lauréate en avril 2024 du prestigieux prix Baillet-Latour biomédical. Son thème de recherche se base sur le renforcement d'efficacité de l'immunothérapie des cancers et c'est ce dont elle va nous parler maintenant.
Dear colleagues, it's such a pleasure for me to be here today and to present you our work. And today I'm going to talk about our recent discovery, which is about targeting the adrenergic receptor to increase the efficacy of cancer immunotherapy. The principle of immunotherapy is based on the notion that cancer cells, they can present specific antigens. And the CD8 T lymphocytes, they can recognize these antigens and attack tumor cells and kill them. And soon after this discovery, we have realized that this process is more than complicated. In the environment of the tumor, there are different immune cells can help T cells to do this function, including dendritic cells, anti-tumor macrophages. And also, there are cells that can inhibit this process. This includes pro-tumor macrophages, myeloid-derived suppressor cells, and regulatory T lymphocytes, and etc. In recent years, it has been discovered that tumor cells can express certain molecules on their surface, and these molecules can directly bind to some receptors on the T lymphocytes or other immune cells, and directly shut down the function of these cells. So current immunotherapy strategies are focusing on two arms. On one hand, to increasing the T cell function to kill the tumor, to improving these functions. On the other arm, to deblock all the suppressive mechanisms that are preventing the T cell to do so. One success is the immune checkpoint blockade therapies. For example, the anti-PD-1 and anti-CTLA-4, and this has shown very promising clinical results across various tumor types. With advanced metastatic myeloma as an example, as compared to the chemotherapy in the past, which can only result in around 15% survival rate, the anti-CTLA-4 and anti-PD-1 therapy were able to bring up the survival rate to up to around 50%. This is really promising and telling us by modulating our immune system, we might be able to eliminate the tumor in a more specific way. However, even with this example, which is one of the best responding tumor type, there are still a lot of patients not responding, the other 50%. And it is important to note that in other type of tumors, the responding rate is much lower. The majority of patients are still not benefiting from the therapy. So for more patients to benefit from immunotherapy, the key is to discover new therapeutic targets, like the PD-1 and CTLA-4, and bring them to clinic. That is actually the research focus of my team. And today I'm going to present you a new immunotherapeutic target we have discovered recently in our lab, which is called alpha-2 adrenergic receptor. And this receptor is a GPCR and belongs to the adrenergic receptor family. It's one of the three receptors, beta, alpha-1, and alpha-2. Their major function actually normally is to bind to and to be stimulated by adrenaline and noradrenaline, and thereby modulating the smooth muscle contraction, cardiac function, and also blood pressure homeostasis. And the agonists and the antagonists have been developed and already used in clinic to be used to manage cardiovascular disease disorders and also to treat hypertension, for example. However, actually, so far there has never been a link described between this receptor and cancer immunotherapy. And how did we come to this receptor? Actually, we came to this receptor because at the time we were working with a compound called guanabans. And we were interested to know how the tumor's stressful environment is modulating different immune cells, thereby modulating their function. And this compound is a modulator of the stress pathway. And we were curious to know if we give this compound into the tumor-bearing mice to modulating the different immune cell response to the stress, will we have a better therapeutic response? And yes, we did. As you can see, in the MC38 colon cancer model, for example, treatment with guanabans resulted in a very nice tumor inhibition. That is good news. However, soon after, we have excluded the hypothesis of the pathway involved. We have shown that this anti-tumor effect is not dependent on the pathway where we were studying at the time, the integrated stress pathway. So we started to look for other mechanisms or targets. And since guanabans is also widely known as an agonist of alpha-2 adrenergic receptor, we quickly tested two other agonists like guanabans to see if they would behave in the same way. We tested clonidine and guanfacine. And we have seen a similar anti-tumor effect with all three agonists tests at the time. That was encouraging. And just to note, these are monotherapies without combination with any other type of therapies. And then we were curious to know if these compounds are only working on colon cancer, because there was never a link described between this agonist and anti-tumor effect, or it is a general effect. We quickly tested several other tumor models, including hepatocellular carcinoma. As we can see, both compounds also yield at a very nice anti-tumor response. T-cell lymphoma, lung carcinoma, and myeloma, breast carcinoma, renal carcinoma, and fibrosarcoma. And with all the models we tested at the time, and there was always an anti-tumor response. Although the response was greater in some models and less effective in some models like breast cancer, however, we always found anti-tumor response. So the tumor growth inhibition by this agonist is observed across various types of tumors. There's no tumor type selective effect. And this actually led us to think this agonist might not work directly on tumor cells, because how can a compound target all the different types of tumor cells? Rather, it might work on something that is common, and as a lab study immunotherapy, we thought about immune systems. So we designed an experiment to test whether these agonists are inhibiting the tumor growth by directly targeting tumor cells, or via their impact on the immune system. So we implanted the B16F1 melanoma cells in parallel to either immunocompetent mice, meaning the mice have intact immune system, or immune deficient mice. And these mice do not have a sufficient immune system So they cannot have a proper T cell or NK cell response. As you can see, mice treated with guanabans have, in the immune-competent mice, had a very nice tumor inhibition. Measurement of the T cell infiltration as a hallmark of the immune response resulted in a significant increase of T cells, CD8 T cells in the tumor. And this inhibition was not seen in the mice that do not have an intact immune system. And same was found for other agonists, such as clonidine. In the immune-competent mice, there are inhibition, but in the immune-deficient mice, there's no inhibition. So this is actually, in a way, proven our hypothesis. These compounds are working on our immune system, so that that is why all the different types of tumor could be inhibited by these compounds. As I mentioned in the previous slides, one important issue to solve right now for cancer immunotherapy actually is to address non-respondence in those patients who are not yet benefiting from the anti-PD-1 or CTLA-4 with therapy. And to know whether these targets could be used to overcome this barrier, we tested their anti-tumor efficacy in models that we know that are not responding for this anti-PD-1 or CTLA-4 therapy. One model we tested is a transgenic mouse model we developed in our lab. We call it TIRT model. It's a melanoma model. The tumor formation will be induced by tamoxifen and it is formed spontaneously, quite mimicking the patient situation. And as we can see in this model, the tumors are not responding at all to anti-PD-1, anti-CTLA-4 therapy. Actually, these tumors are not responding to any type of immunotherapy so far tested in our lab. However, when we give this model, these mice guanabans, the agonists, there was a very nice tumor inhibition. And actually, there is a slight, even slight better response when we combine the three. So this was very encouraging, and we quickly tested other models that are also known to be resistant to the PD-1 therapy, like TC1 lung cancer. Again, as you can see, anti-PD-1 has absolutely no anti-tumor effect. But clonidine itself already, as a monotherapy, already has a very nice anti-tumor effect. Combined with PD-1 has a greater effect, but it's minimum. So this is very promising because this means that this compound potentially can be used to give to patients which are not responding to the current available immunotherapies. So far, all the data are about mouse models and mice immune system, but the important issue to solve is to know whether all these findings can be translated to human. In the laboratory, however, we cannot directly working on the patients, and we have different kind of models to test. One model is human xenograft mouse model. So we quickly decided to test this compound in these models. So how do these models work? So we would transplant or let's say inject to human tumor cell line, such as ovarian carcinoma scope 3, into immunodeficient mice. Why? Because in this case, only in this way, the mice immune system will not reject the tumor. And then when the tumor appears, we would give the mice allogenic human-derived PBMC, and this setting will trigger allogenic anti-tumor response from the PBMC towards the tumor cells. And at the same time, when we give the human PBMC, we give the mice big injection of the agonists. As you can see, if we only give the mice agonists but without the PBMC, there is no effect because there's no immune system to mediate this anti-tumor effect. And if we only give the mice human immune cells but no compounds, there's also no effect. It means the natural allogenic reaction was not strong enough to inhibit the tumor growth. However, if we now give the compound together with the human PBMC, they have a very nice tumor inhibition. So although this system is not really the same as what is in the patient, it is very promising. It means that this compound potentially can regulate the human immune system to result in a tumor inhibition. We also tested the infiltration of human T cells into the tumor, and there is a significant increase of T cells. And same was found for other agonists, such as clonidine and also guanfacine, and also they also resulted in tumor inhibition and increased T cell infiltration. So after we obtained all these results, important issue to solve is that are all these kind of nice tumor inhibition results mediated really by this target, because molecules can target many things and we were not 100% sure about it. And to answer this question, we first decided to use UCLouvain, which is an alpha-2 adrenergic receptor antagonist. And to know if we block this activation of this receptor while giving the agonist, so agonist cannot be so efficiently bind to receptor, can we reverse this tumor inhibition? Again, we tested in the humanized model, the xenograft model, by giving a nice human tumor cells and PBMC, and give the combination of UCLouvain and agonist. Again, as you can see, the agonist result in a very nice tumor inhibition when given together with human immune cells. And UCLouvain, the antagonist, completely blocked this anti-tumor effect, proposing that this alpha-2 adrenergic receptor is the target of the agonist. To have a final proof and validation of this receptor to be the target that is mediating this anti-tumor effect, we decided to give the knockout mice. We tested the anti-tumor effect in the in the wild-type mice or in the receptor knockout mice. As we can see, in the wild-type mice, both agonists have resulted in a nice anti-tumor effect, and also there is increased T cell infiltration. And the anti-tumor effect by these agonists are completely abolished in the receptor knockout mice. Same was for the T cell infiltration. So this has given us a final proof that this receptor is the target that is mediating this anti-tumor effect. So, after obtaining all these promising results, the important question to answer, and we are aware there's a long way to go, is to understand the mechanism of action of this agonist. How do they modulate our immune system to attack the tumor cells? The first obvious mechanism why one could think is their impact on CD8 T cells, because there was increased CD8 infiltration, and if they make the T cell better, maybe that's the way they attack the tumor better. So we decided to test this hypothesis. We compared the tumor growth inhibition in mice that are normal, there is a nice inhibition, or in mice that are depleted for CD8 T cells. So we injected antibody to deplete these T cells. As you can see here, the nice anti-tumor effect that is caused by the clonidine is abolished when we deplete CD8 T cells in these mice. So this is meaning that the tumor growth inhibition by these receptor agonists is partially mediated by the CD8 T cells. However, it doesn't mean that the anti-tumor effect is a result of this agonist on the CD8, and then that's a direct modulation. To answer this question, we use the following experimental setting. As you remember, there was no anti-tumor effect observed for the agonist when the agonist was given when the mice had no receptor, no alpha-2 AR receptor, like this. Then we decided if we now transfer wild-type CD8 T cell into these mice, the CD8 T cells will be the only cell types that will express this receptor. And now if we give the mice the agonist, the anti-tumor response should be improved, because if the hypothesis is that they can improve the T-cell response, that should be the result. However, we did not really see an improved anti-tumor effect after we gave the wild-type CD8 T-cells, as we can see. When we transfer CD8 T-cell, there was already an anti-tumor response, but the cloning did not further improve it. This is indicating that CD8 T-cells is not the direct target of this agonist. And so what other cell types can mediate this anti-tumor effect? The second cell type we checked was myeloid-derived suppressor cells. The cell I introduced in the beginning that is blocking the CD8 T-cell function. And why were we so interested in this cell population? As I mentioned a while ago about this transgenic mouse model, T melanoma, that's in our lab, that we have shown that they are completely resistant to all types of immunotherapy. A couple years ago, we have discovered that one of the reasons why this model is so resistant to immunotherapy was because they were highly infiltrated with MDSCs, myeloid-derived suppressor cells. And these MDSCs, they can delete CD8 T cell in the tumor by inducing their apoptosis. Therefore, no matter which therapy you use, the T cell cannot survive. And since one of them has a very nice anti-tumor effect in this model, we thought about maybe they could impact MDSCs, thereby release the anti-tumor effect of CD8 T cells. And we first measured the MDSC infiltration in this model after the treatment. As we can see, with guanine treatment, there was indeed a significant reduction of these suppressor cells. So when we treat mice with this agonist, these cells are no longer there, so they cannot suppress the immune response. And indeed, when we measure CD8 T cells, because the MDSCs are removed, there is an increased infiltration of the tumor-specific CD8 T cells. As we hypothesized, these T cells are less apoptotic because MDSCs are gone. They cannot induce apoptosis anymore. So these T cells are less apoptotic and they function better. This is measured by measuring the electron gamma production by the CD8 T cells in the tumor. So all this result is indicating that this alpha-2 agonists, they can reduce MDSC infiltration and the suppression caused by these cells in the tumor model. However, as I showed you before, this tumor model is one example that is highly infiltrated by MDSC, but across all those tumor cells that we studied, that they are showing nice tumor inhibition, not all the tumors are highly infiltrated by MDSC. It's hard to believe this will be the only reason that all these models are responding to the agonists. So besides MDSC, what other cells are impacted by the agonists? To answer this complicated question, we decided to perform a single-cell sequencing analysis of the cells in the tumor microenvironment of mice treated with a control vehicle, always cloning. As you can see, these dots are showing the different immune cell type present in the tumor of the mice are treated with control or treated with cloning. As you can see, we can find tumor cells, CD4 T cells, CD8 T cells, neutrophils, and the majority of cells appear in this model was apparently macrophages. And based on their signature, we further subgroup the macrophages into 5 different groups: 1, 2, 3, 4, 5, because macrophage has a very continuous different kind of phenotype in the tumor. And after doing so, we compare the different cell composition in mice treated with a vehicle or with cloning. As you can see, besides increased CD8 T cells and reduced tumor cells, as we shown before in our experiments, The cell type that is appeared dramatically changed is the macrophage. If you agree with me, the macrophage streak, which was very abundantly present in the control mice, disappeared in cloning treated mice, and the macrophage 1 group, which represents the most active macrophage subgroup, is now abundant after cloning treatment in the tumor. This has drawn our attention to the effect of the agonists on macrophages. We did some experiments to verify these effects. We analyzed the macrophage phenotype in the macrophages that are in the tumor in this same tumor model, anti-UCLouvain model, with or without cloning treatment. We use 2 assays to measure their activity. We measure their phagocytic activity, which is the ability how the macrophage can pick up the tumor cell debris or even tumor cells. And another assay was to measure their surface expression of NBC class 2, which is a molecule presenting the tumor antigens to T cells or other immune cells after they pick up the tumor cells. As you can see, in the wild-type mice, the cloning treatment dramatically increased the phagocytic activity of these macrophages, and also there is a higher expression of NC class 2, meaning that this treatment with this agonist can increase the macrophages to take up tumor cells and transfer the antigen formation to other immune cells. And this effect was completely abolished when we tested in the knockout mice. So we could confirm the single-cell sequencing results that macrophage is a major target of the agonists. We further performed experiment to verify whether this the causal link relationship is true. First, as similar as we did for CD T cells, we decided to decrease macrophages to see if the macrophages are not there with the anti-tumor effect still present. We used the liposome clonate to decrease macrophages. As we can see, in the control group, there is a nice tumor inhibition by cloning. But if we remove macrophages by the liposome clonate, the tumor inhibition effect is milder, proposing that macrophage is involved in the anti-tumor effect of the agonists. The other way around, which is more important, is to understand do agonists directly impact on macrophages and thereby stimulating anti-tumor immune response. To do so, again, we use the alpha-2A mice, which the agonists do not work. We have injected this tumor now with wild-type macrophages, so the macrophage will be the only cell type expressing the receptor in the mice. And then if macrophage is the cell responsible for the response, now the agonist should work. And indeed, when we give the knockout mice the wild-type macrophages, and we have seen that the cloning is again able to inhibit the tumor growth. Actually, same was observed for the T cell infiltration. In the knockout mice, there is no increase in T cell infiltration, but when we give the wild-type macrophages, the T cell infiltration increase is again observed. These results are proving that at least the agonists are able to modulate the macrophage function and thereby promoting anti-tumor response. So altogether, our results so far is indicating that alpha-2 AR agonists they can bind to anti-tumor macrophages and myeloid-derived suppressor cells. On one hand, by binding to the IDSCs, they remove this suppressive cell from the microenvironment. On the other hand, by binding to the anti-tumor macrophages, they stimulate the anti-tumor macrophage function, which in turn stimulates CD8 T cell function and lead to the tumor cell death. So, as a final conclusion, and I think it's obvious, I hope I convince you that alpha-2 agonists, they have shown great potential in immune-mediated anti-tumor activity, even at the monotherapy. And they act as these are myeloid cells, macrophage and BSCs, and they could be used to improve the efficacy of cancer immunotherapy in patients potentially. But there's a long way to go because I think this study opened the door for us to answer all many questions. For example, for the understanding of the underlying mechanism driving the anti-tumor activity of the agonists, which signaling pathways are involved, are cells involved, or secondary and inferior organs involved. And also it brings some new ideas about crosstalk between nervous system and immune system. With this, I will finish my presentation, and I thank you for your patience and listening.
Thank you very much. There is a question in the room. Jean-Michel,
Oui. Thank you very much for this nice talk and very impressive results. We are not talking about the concentration of the agonist used. Is there a therapeutic level, or is it at the lower concentration and the therapeutic level used? Because there are quite side effects on those compounds.
That's true. The compound we used was 5 milligrams per kilogram, which is at a higher dose than what is usually used for patients to treat hypertension. The reason is because we think that the metabolism of this compound is very quick. Actually, when you eat the mice, it's quickly activated. And that is why we think there are room to improve to really, refine this therapeutic strategy to see how to bring innovative approaches to make it usable for patients.
Thank you very much. So does that mean that the anti-PD-1 refractory tumors may be so because of a lack of anti-tumor macrophages? I mean that it's not because T cells do not become responsive, but because you need pro-tumor anti-tumor macrophages?
I think there is an element of truth in it, because I think a macrophage is one of the most easily treated immune cell type in the tumor. And it is a very important factor to produce long, durable response. And with our results, we are actually very interested in the macrophage role in the anti-tumor effect. And whether the anti-PD-1 will not work because there is no modulation on macrophages, this I cannot really answer this question. I'm not 100% sure, but I think this is a direction we could explore. On the other hand, I think, like I said, there's a long way to go to understand how this compound really working, because macrophage might be one element, but I believe to produce a long, durable response, you need a very nice, coordination throughout the body. They need to have the bone marrow continue to produce and differentiate correct immune cells, and also we need a secondary lymphoid organs to have a correct thing happening there, and finally the whole machinery has to work continuously. That is what I think these compounds are doing. And actually with our results that are not in the publication, we know that these receptors are highly expressed in the bone marrow and also in the secondary lymphoid organs. And we also know if we block the trafficking between the secondary lymphoid organs and the tumor, actually the response is greatly abolished. So for me, one factor that could lead to this nice effect is actually what is nice to explore is to understand how these compounds are coordinating this whole process. Not only in the tumor macrophage, not only on the macrophage in the tumor, but rather from the whole generation of the whole macrophage compartment, to make it into a very effective tool. When they enter into the tumor, they can really have a durable response. Yes.
And second question: do you think that this therapy may help use CAR T cell therapy in solid tumors, which is for the moment not working, if I understand?
Yeah, I think there is a good chance because in our study we have combined T cell therapy, so adopted T cell therapy with this compound, and we have seen that indeed it synergizes with T cell therapy, so you can improve it. On one hand, you will remove the suppressor cells. On the other hand, you will increase macrophage activity. Lastly, which is actually we are currently exploring, we are really interested to know if this compound are really making the trafficking of the immune cells to the tumor much easier. Because as we know, the smooth this compound, the receptors expressed on the smooth muscle cells, and lymphatic vessels and blood vessels are surrounded by this cell type. And this is a factor I really would like to explore, to know if that is really making the trafficking much better. If that is so, indeed the CAR T therapy will be improved.
Michel Benoît, Stéphanie Constantinescu.
Well, we are ready to congratulate you for what is a real breakthrough in tumor immunotherapy. Now, I'm wondering about other fields, especially infectious diseases, in which pathogens live within macrophages and in which CD8 T cells play an important role. I'm thinking, for example, about tuberculosis. And I'm wondering whether you had an opportunity to test your compound in another model of infectious disease with intracellular pathogens.
Yeah, that's a really nice angle to explore. Unfortunately, we do not really have the models, and we haven't tested yet. But if any laboratory would have these models available, I think it will be really interesting for us to test it. Yes.
Congratulations for a very, very clear presentation and extremely important work. I found out that anesthesiologists, when they participate in orthopedic surgery, which is very painful after, they have a protocol to inject clonidine in the bone, and that reduces inflammation and pain afterwards. Do you have any evidence of anti-inflammatory effect of your agonists that could actually prevent maybe secretion of some proteins that could disfavor the immune response?
I do not have evidence about that. If you talk about the bone marrow cells, the only thing we have observed was we know there was a modulation of STAT3 and STAT5 pathway if you give mice this compound. And if you give the bone marrow to do Western blocking, and the STAT5 or STAT3 pathway is modulated. With STAT5, I think it goes up. And STAT3, I think STAT3 was also going up, yes. But for the other things, we did not test it yet.
Before continuing in this room, we will give
The floor now to internet. And guess who's there? Professor Lamy.
Yeah.
Professor Lamy.
My first question was related to the one Dr. Kaufmann was asking about the dose of clonidine used and the potential side effects, like, for instance, hypertension. And my second question was related to another alpha-2 agonist extensively used by anesthesiologists, either in human practice or in animal practice, which is dexmedetomidine. So have you some experience with this other one, alpha-2 agonist, dexmedetomidine?
Okay, so for the first question about side effects, actually this was a question I received each time when I mentioned this agonist. Yes, this agonist, they have some sedative effect because it will go via the central nervous system. We talked to some specialists in this field, and in his opinion, these side effects are manageable, even also with the blood pressures, which are manageable in clinical practice. And also, since sedative effect and many other effects are working through the central nervous system, as I mentioned before, maybe the strategy could be refined, for example, to develop compounds that are no longer reaching there. So, I think there are some ways to try to refine to reduce the side effect while maintaining the therapeutic response. Second question is about the other agonist of the receptor. Yes, I'm aware of this compound. Unfortunately, we have not tested this compound in vivo. The reason was just because I think at the time it was not available as high amount and as a good how to say, budget for us to test it. But I think that is something we should test because there are some data on that compound more than cloning. So that's something we will do. Thank you.
Thank you so much, and congratulations.
Thank you.
Thank you. Jean-Michel Fauder?
Yes, congratulations for this really outstanding lessons of biology and immunology. It's really an outstanding basic science, and you show that we really deserve this outstanding prize of the Baillet-Latour. Congratulations. But you know that epidemiologists can all the time in the clinics tend to correlate all kinds of factors. And since the alpha-2 agonists are widely, and the alpha-2 blockers are also very widely used in the clinics for decades, if there were any correlation between the use of those drugs and the immune control of tumor development, it would probably have been disclosed already. Are you aware of any study that would demonstrate in the human clinics a correlation between the use of those drugs and tumor deaths and tumor growth?
Unfortunately, I haven't seen any study studying it, even in clinical trials or something. Also, we actually, when we did the study, we were really interested to know if there are database or that we can use, even though single cell or bulk RNA-seq, that we can correlate some data to give some clue. Unfortunately, these drugs were not given anymore for hypertension for a while, so we were not able to obtain these results. But if you are asking about some clinical relevance, I think I can answer the question with another example, actually the beta blocker, the beta adrenergic receptor. The beta adrenergic receptor is actually, I wouldn't say completely opposite, but it goes to a different direction than alpha-2. Beta receptor is linked to GES, and alpha receptor linked to GI. Why is they are opposite? And the beta blocker is going to reduce blood pressure. Alpha agonist is going to reduce the blood pressure, right? So although alpha-2, there was no available evidence, but for beta there are actually. There are actually already clinical studies performed with beta blocker that have shown that if you give patients the beta blocker, there is a nice better survival, patient survival. And also, there are also evidence to show that in cancer patients, there is a higher level of adrenaline. And the study has shown that this might contribute to the tumor progression. There's a correlation about the adrenaline level and the tumor progression. So that is why I think beyond this receptor that we are working on, the whole neuron and immune crosstalk is really something we should go further to explore, which might give us more ideas and clues. Thank you.
Last question. You have a question? No? Okay. Thank you. Thank you very much.